inside every single one of our cells, there's a language being spoken, a secret code that literally decides the life and death of individual proteins. And for the longest time, we thought we had it all figured out. Turns out, we were wrong. So today, let's decode the ubiquitin code, because what we're finding is a system of just staggering complexity and power.
Okay, so let's start with what you probably learned in a textbook. For decades, the story was super simple. Ubiquitination is just the cell's trash tag. It's a molecular death sentence, right? It marks a protein for destruction. But is that really all it is? Is this just a fancy garbage disposal system? Or have we been missing a much more sophisticated code that's running pretty much everything inside the cell?
All right, let's start peeling back the layers here. We're moving way beyond that simple trash tag idea. At its core, ubiquitination is what we call a post-translational modification, which, is really just a fancy way of saying it's an edit made to a protein after it's already been built. And this particular edit, well, it's a dynamic, reversible signal that does so much more than just take out the cellular trash.
I mean, the sheer scope of its influence is just, wow, it's staggering. It's not just about deciding which proteins get to live or die. No, it's a master regulator of signal transduction. That's how our cells talk to each other. It's a critical player in DNA repair, fixing damage before it can turn into a nasty mutation. It's directing our immune responses, and even guiding how a stem cell decides to become, say, a neuron or a muscle cell. It's this fundamental language the cell is using to manage its entire operation.
So how does the cell actually attach this incredibly important tag? Well, it's not just a single event. It's a highly coordinated three-step process, an enzymatic cascade. The best way to think about it is like a molecular relay race, where the ubiquitin molecule is the baton, and it's getting passed from one enzyme to the next with just unbelievable precision.
Let's break down that relay. First up, you have the E1 enzyme. It uses energy, from ATP, to activate a ubiquitin molecule. Simple enough. Next, that activated ubiquitin gets handed off to an E2 enzyme, the conjugating enzyme. And then, in what is absolutely the most critical step, the E3 ligase enzyme grabs that ubiquitin from E2 and attaches it to the specific protein it's meant for. So E1, E2, E3, activation, conjugation, and transfer.
Okay, now let's focus on the real star of the show, because while E1 and E2 are totally essential, the true architect of this whole process, it's the E3 ligase. This is the enzyme that makes the big decision. Which protein gets tagged, and when does it happen? Pretty much all the specificity of the entire system boils down to this one player.
So the E3 ligase, it acts like a molecular matchmaker. It's got this unique ability to recognize and grab onto a specific target protein on one side and the E2 ubiquitin complex on the other. It physically brings them together, making sure that ubiquitin tag gets delivered to the exact right address every single time.
And the system has just incredible diversity. We're talking about hundreds of different E3 ligases, and we can group them into these big classes like ring, hect, and RBR ligases. Each one uses a slightly different mechanism, and that diversity is exactly what allows the cell to regulate so many different things at once. But this brings up an even deeper question. If the tag is always the same molecule, ubiquitin, how does the cell know what to do with it?
And this is where it gets really, really cool. We've been talking about how the tag gets put on. But the real secret, the real language, is in the tag itself. It turns out it's not just that a protein gets tagged with ubiquitin, it's how it gets tagged. The actual structure of the ubiquitin chain forms a distinct message, and that message dictates a very specific outcome for the protein.
And this slide just illustrates the power of this code perfectly. It's amazing. On one hand, you've got a K48 linked chain. This is the one we all learned about, right? The classic signal for destruction. It sends the protein straight to the proteasome to be chewed up. But then you have the K63 linked chain, and it does something completely different. It's not a death sentence at all. It's a call to action. It builds these scaffolds for signaling complexes and recruits other proteins for things like DNA repair. Same molecule, different linkage, a totally different fate.
And that right there, that's the key takeaway. The incredible complexity, and frankly, the power of this entire system lies in the how. The specific connection point used to build the chain is what determines the function. It transforms a simple tag into a sophisticated language that directs all the machinery in the cell.
So what happens when this precise, elegant code gets misread or just breaks down? Well, as you can probably guess, the consequences of this kind of dysregulation are severe. They actually form the molecular basis for some of our most challenging and devastating diseases.
Yeah, when the ubiquitination system fails, the results can be catastrophic.
Errors in this pathway are directly linked to the uncontrolled cell growth that we see in cancer. They're also implicated in that toxic pileup of proteins that leads to neurodegenerative disorders like Alzheimer's and Parkinson's, and even the chronic inflammation that drives autoimmune diseases.
Let's look at a really concrete example. There's an E3 ligase known as the APCC complex. Its job is to degrade these proteins called cyclins to make sure the cell cycle moves along properly. Now, if this E3 ligase fails, those cyclins build up, the cell cycle checkpoints get ignored, and what do you get? Uncontrolled cell division. That's the hallmark of cancer. It's a straight line from one tiny molecular error to a devastating disease.
So this all brings us to a really critical challenge for researchers. I mean, given this incredible complexity, how do you actually study it? How do you read the code accurately without just getting lost in all the noise? To really dissect these pathways, you need a level of precision that just goes way beyond the standard methods.
The nuance here is absolutely everything. A standard assay might tell you, yep, this protein is ubiquitinated, but it'll probably miss the crucial linkage-specific details. Is it a K48 or a K63 linkage? I mean, that's the difference between a degradation signal and a signaling scaffold. Just blindly sequencing everything is super inefficient. To get real, meaningful answers, you need a targeted strategy, one that can enrich for these fleeting, low-abundance modifications, identify the specific linkages, and then quantify the changes accurately.
You know, this quote just perfectly captures the essence of everything we've talked about. In the incredibly complex world of the cell, a single ubiquitin molecule, that might just be a signal, but the way it's linked into a chain, that transforms it into a full sentence with a clear, specific command.
So, let's end with this question for you to think about. As you go out and tackle your own biological questions, just remember the insane complexity that's hidden in this system. Are you really equipped to see beyond the tag, to distinguish the signal from the sentence, and to truly read the ubiquitin code? Thanks for joining me.